Good day, I'm Romelyn BAPTISTA, and I will be presenting my study entitled In Silico de Novo Synthesis and Structure-Based Design of PS273R Protease Inhibitor as Potential African Swine Fever Therapeutics, a Molecular Docking Study. First, I'll start with a brief introduction. What is African swine fever? African swine fever is a viral hemorrhagic disease that affects domestic pigs and wild boars. It is caused by the ASFV. It is characterized by anorexia, bloody diarrhea, vomiting, and high fever. Most recently, ASFV emerged in the Philippines, and because this virus is novel, the control measures rely strictly on sanitary measures, resulting to serious economic and production losses. Vaccine and antiviral drugs are believed to be the best tools for prevention and control of ASF. Unfortunately, all the attempts to develop an effective vaccines had failed. Therefore, it is in great need to develop effective antiviral drugs. So the antiviral drugs can be classified into two groups. In this study, small peptide inhibitor was chosen because peptides in particular present several advantages, as shown here in this slide. So peptides represent an excellent starting point for the targeted inhibitor design of the lead molecules. In this study, the target protein will be the viral protease PS273R. Why do we need to target this protein? This is because the gene expression of ASFV was discovered to be similar with other large DNA viruses, such that it encodes two large polyproteins precursor, PP220 and PP62, which are proteolytically cleaved by their own viral protease to generate the structural proteins required for virus assembly, morphogenesis, and infectivity. Therefore, the PS273R protease is believed to be an attractive target. So shown here is the structure of the protease, and the box part is the active site of the protease. So the general objective of the study is to develop a de novo drug design that can be utilized to treat ASF primarily by inhibiting the enzymatic activity of PS273R using a peptide inhibitor. To demonstrate, shown here is a structure of the protein. If a natural ligand binds to the protein, they will be proteolytically cleaved to generate the mature proteins required for replication and morphogenesis. However, if a peptide inhibitor binds to the receptor protein instead of the natural ligands, the proteolytic processing of the PS273R will be blocked, ultimately inhibiting the ability of the ASF genome to produce infectious virus particles. So here is the overview of my study. First, the protein preparation will be done using thymol. The PDB file of the protein and the binding site coordinates will serve as input in the de novo synthesis using ELID. From the generated peptide inhibitors, only the top 5 molecules will be selected and visualized. The visualization of the protein peptide complexes will be done using thymol and lig plot. Take note that the crystallographic structure of PS273R in RCSV is not bound to its natural ligand. Hence, there is no control molecule provided. To overcome that, the molecular structure of polyproteins were predicted using AlphaFold. So the 6 control molecules and the 10 lead molecules will be subjected to molecular docking using HPEP-dock. Only the control and lead molecules with the highest docking scores will be further analyzed using MD simulations, amyloidogenicity, and antigenicity. So moving on, the results of the automated de novo drug design of ligands are found in Table 1. As shown in the table, the docking scores provided are negatives, indicating favorable binding. Take note that the more negative the score, the better. Therefore, only the top 5 molecules generated with a more negative docking scores were considered for further analysis. As you can see, generation 20 showed the most negative plant scores of negative 100.25, indicating that this lead candidate has the best docking conformation with PS273R protease. The second highest ranking molecule is G18 with negative 100.16 docking score. The third highest ranking molecule is G17, the fourth is G14, and the last is G16. So next, the sequence of polyproteins acquired from the UniProt database were subjected to structure prediction using AlphaFold. Figure 6 to 8 shows the predicted structures of polyproteins, and the AlphaFold results are summarized in Table 2. So each predicted model was ranked based on their PLDDT. The highest accuracy cutoff is PLDDT greater than 90 and 70. Using this criterion, models 1 of PP62 and PP220 showed the highest score of 81.26 and 70 respectively, indicating that these models have the most accurate structure prediction. So shown in this figure are the sequences of the cleavage sites of the polyproteins. So notice that the protease identifies the second gly residue as a common recognition signal for cleavage. This serves as the guide in the extraction of cleavage sites. So from the predicted models, the structure of the 6 cleavage site were extracted and used as the positive controls for the molecular docking analysis. So next, in this study, the visualization was done using PyMol and LigPlot to know which residues of the peptide molecule interacts with the binding site residues of the receptor protein. So figures 9 and 10 summarizes the visualization done for G20. As shown in figure 10, 5 polar contacts represented by this yellow dashed line exist between the interacting residues of PS273R and G20. The interacting residues were also summarized in Table 4. So the 2D diagram from LigPlot also shows the residues involved in the hydrophobic interactions. So similarly, in the DAC complex of G18 molecule, 5 polar contacts were observed. The interacting residues were also summarized in Table 6. So the rank 3 to 5 ligand molecules exhibit similar hydrogen bonding and hydrophobic interactions as that of the rank 1 and rank 2 molecules. So overall, the key residues observed are the hydrogen bonding interactions of proline 193, asparagine 187, threonine 159, and glycine 166. So next, the peptide screening technique using SAT-PDB database was done because the de novo synthesis of peptide is very difficult. However, it showed that not one therapeutic peptide has similar structural fragments as that of the de novo peptide. So to overcome this drawback, the leads were drawn using PyMol based on the structural information of the de novo peptides, side chains of amino acid, and of course, the visualized atomistic interactions that I have discussed earlier. From there, 10 lead molecules were drawn, as shown in figure 17 to 26. So molecular docking was done to further confirm if the John ligands have more favorable binding conformation than the control molecules. So the rankings for the top 10 binding models were provided together with their docking energy scores as shown in Table 7 and Table 8. From the 10 lead molecules, LM1 showed the highest docking score of negative 170.532, while control molecule 3 showed the highest docking score of negative 148.07. Comparing the results, LM1 have better ligand conformation because of its relatively lower docking energy score compared to CM3. This indicates that LM1 have a more favorable binding affinity towards PS273R. So the LM1 and CM3 complex were visualized using PyMol and LigPlot to confirm if they exhibit similar interactions as that of the synthesized molecules from E-lead. As shown in figure 29 and table 10, CM3 exhibits similar hydrogen bonding interactions with the ligand molecules from E-lead, specifically the polar contacts with glycine 166. Similarly, as shown in figure 31 and table 12, LM1 also featured some key binding interactions for PS273R. Overall, results showed that the original ligand and the John ligand mainly interacts with PS273R through hydrogen bonding interactions with asparagine 187, threonine 159, and glycine 166. The similar interactions observed indicates that the John ligands are likely to have the same molecular structure and sequence with the lead molecules obtained from the E-lead. So the molecular dynamic simulation using GROMACS was done to account for the changes of the atomistic behavior, stability and flexibility of the molecular system upon peptide binding. So here is the visualization of the trajectory file obtained after MD simulation using VMD. So first, the potential energies as a result of the energy minimization were obtained. As shown in the plot, the potential energies of the systems are both negative. This steady convergence at low potential energies indicate that the system is stable and at energy minimum. This ensures that the structure of the protein-ligand complex is relaxed, there are no steric clashes or inappropriate geometry observed. So next, the RMSD was computed to measure how much the protein structure changed during the simulation. So as shown in the plot, at about 0.5 nanoseconds, the RMSD levels off around 0.1 to 0.15 nanometer, which is quite small, suggesting that the systems are stable. From there, the RMSD levels ranges only from 0.1 to 0.2 nanometer. However, at about 9 to 10 nanoseconds, the RMSD increases to 0.25 nanometer, which is slightly higher than the optimal RMSD value of 2 nanometer. So this result is actually anticipated as the protein might still move even though position constraints were established during equilibration. Regardless of the observed minor movement, the overall conformation was still maintained. The RMSD of both equilibrated systems with respect to the crystal structure were also computed. You can see that they are not exactly matched and there are subtle differences. Again, this is to be expected since both systems undergone energy minimization and because the position restraints are not completely perfect. Regardless, both RMSD plots still levels off around 2 nanometer, suggesting that the binding conformation were stable during simulation. The next is the RMSF. So the RMSF stands for root mean square fluctuation, and it calculates how much a particular residue fluctuates during a simulation. As shown in the plot, the RMSF of most residues at the binding site was below 0.3 nanometer, indicating that both the ligands kept close contact with their binding packets during MD simulation. In the case of CM3, which is the green one, the highest fluctuations were observed between the 4,000 to 4,500 atom stretch, whereas LM1 exhibits highest fluctuation between 0 to 500 and also in the 4,000 to 4,500 atom stretch. Still, the RMS of most residues is found to be stable below 0.3 nanometer. Lastly, the radius of variation of the system was shown in figure 40. So the RG values remain relatively steady, suggesting that the compactness of the protein-ligand complex is very stable over the course of 10 nanosecond simulations, and that the ligand molecules has not significantly interfered with the folding mechanism of the protein. So next is the amyloidogenicity analysis. It was done using amylogram to analyze if the peptide sequence exhibits amyloidogenic properties. This is because amyloids are misfolded protein aggregates that exhibit very high mortality and cytotoxicity. So from the graph, the probability of the amino acid sequence to self-assemble as amyloid aggregate is only 0.3, which is lower than the cutoff value of 0.5, indicating that LM1 doesn't exhibit amyloidogenic properties. And lastly, antigenicity analysis was done because peptides have the potential to exhibit undesired immunoactivity, which results in the initiation and progression of the disease. The results of the analysis using pro-inflam are shown in table 14. As you can see, the LM1 peptide was predicted as non-pro-inflammatory epitope with a prediction score of 0.5, which is lower than the threshold value of 0.7. In addition, the predicted pro-inflammatory nature of different variants were mostly negative. This result suggests that the basic property of LM1 and its variants are less likely to initiate pro-inflammatory cascade. So in conclusion, this study reports the successful development of a novel and promising antiviral drug candidate for the treatment of African swine fever using in silico methods. Specifically, a peptide of 6 amino acid sequence length was developed to inhibit the enzymatic activity of PS273R protein. The lead molecule 1 showed the highest binding energy score. stable dynamics with the binding site, and good RMSD, RMSF, and RG scores relative to the control molecule 3 after performing the molecular dynamic simulations. Furthermore, LM1 was predicted to have negative amyloidogenicity and antigenicity properties with significantly low prediction scores relative to the threshold values. As such, lead molecule 1 may potentially be a drug lead candidate that can be further modified through other in silico methods to serve as a treatment for ASF. So that is all. Thank you.